Hello, everyone, and welcome to today's live broadcast, Standing Up for the Quality of Our Data with Research Quality Assurance, presented by Dr. Rebecca Davies, Associate Professor at the Department of Veterinary Population Medicine at University of Minnesota. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. We are excited to give you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars. Before we begin, I'd like to remind you that this presentation is interactive. We want to hear from you, so questions and comments could be submitted via the green Q&A button at the lower left of your screen. We'll answer as many questions as we have time for at the end of this presentation. Also, if you are viewing a presentation in the slide window, to enlarge that window, click on the lower right-hand corner icon of the slide window. If you cannot hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button and let us know that you're having a problem. And now, no further ado, I'd like to turn over the presentation to Dr. Rebecca Davies. Welcome.
Talking with you about research quality assurance and hopefully plant a few seeds about new approaches that we can take to stand up for the quality of our data. As Susie mentioned, I'm from the University of Minnesota. I'm the director of a program called Quality Central, where we're working hard to try to integrate quality assurance best practices into our service, training, and research programs. And we're interested in doing this because as research scientists, what we care about are our data. And truly, no matter what kind of data we collect, whether it's in the biomedical field or any other field of study, our data are what are most precious to us and what we need to protect, safeguard, and secure for the life cycle of its development and certainly for the life cycle of our research. So it doesn't really matter whether the data we're collecting, you work at the bench level or whether you work with population data, whether you're interested in generating policy, whether these data are quantitative or qualitative. What's very important to us as scientists is whether these data are protected with a data stewardship approach. And I really believe in the current climate for science that we really need to participate, and I would argue that we need to lead in addressing some of the concerns associated with research reproducibility. And by doing so, by being engaged, it will allow us to stand up for the quality of our work and work to develop sustainable risk-based strategies for ensuring reliable and secure data. So today my remarks are going to be very briefly to discuss some of the threats to research and data quality and then move into strategies we could perhaps consider for addressing threats and strategies that are being considered and being implemented by our stakeholders, those people who are most interested in the data that we generate as part of our research projects. So my remarks again will focus almost completely on whether research quality assurance could be one of hopefully several scientist-driven strategies that we can use to demonstrate and improve data and research quality. So basically, when we think about research data quality, I think you would all agree that there are at least two aspects. One are the sound scientific principles, the heart and soul of our science, the thinking that you folks, you researchers, put in when you design a research project based on a strong research premise. Those types of principles would include things like sound study design, approach to research bias, the statistical models that we select and use to evaluate our data, and our ability to make appropriate and accurate inferences in response to our data. And then the second aspect would be good quality practices. So in other words, we're thinking about what we do, what we decide to do, and how we make those decisions, and also how we do it. What are our practical approaches to generating those research data to answer our critical research questions? So when we think about our research in that way, and we want to consider what are the threats to sound science, to our ability to generate quality research data and make accurate research inferences, I think many of us are familiar with thinking about research fraud down in the tip of this triangle. And of course, research fraud is something that concerns us all, but data have shown that it is a very small contributor to sort of the questions about our research data, 2 to 3%, and I'm sure that's a number we all wish would be smaller, but I think that we need to think broader in terms of data integrity and research integrity. And many people are now spending a lot of time thinking about what are being called questionable research practices. These are areas of our research conduct that perhaps we could do better, and many of us would agree that things like study design, appropriately addressing bias, our statistical methods, are areas that can contribute to research variability, and that certainly might affect the ability of our research to be reproduced. So those are questionable research practices that might be a target for improvement. But then there are also other behaviors associated with how we do the work we do. Simple carelessness, errors that will always occur and that we need to learn from, our routine behaviors, our habits, and the critical influence of mentoring. So as you consider those threats that I recently or just mentioned a second ago, these threats are not described or included in the role of the incentive system, which is when we talk about research reproducibility, many times as scientists we bring up or we consider the role of the incentive system as being a contributor to our problems with research integrity. And my remarks are not going to focus on this role. I'm more interested in looking at how we can secure the data we collect and in our individual and group environments. So I'd like to use this slide just to summarize what I've been saying for the last few moments. This is a table from the World Health Organization guidelines, quality practices in biomedical research, that was provided in around 2006 and is freely available online. And I like this table a lot because I think it summarizes how sound scientific principles and good quality practices contribute to the credibility of our results. And I'll let you look at that for yourself, and I'm sure this is very intuitive and non-controversial, but something we perhaps don't think about enough, that no matter how good our science is, our premise, our hypothesis, our study design, if we don't have good quality practices, we're likely to suffer from that in terms of the credibility of our results. And just as importantly, no matter how good our research conduct practice is, if we are conducting very good practices in the use of research designs that are not good science, the good quality practices won't save the credibility of our research results. So as we think about the necessity of having these two aspects of our research be strong, I'm calling attention to this recent paper by Leonard Friedman and colleagues, Reproducibility 2020, Progress and Priorities. It was just made available online through PLOS, and it really does a nice job of summarizing what some of the issues with data reproducibility or research reproducibility are. And as I looked at that paper and reviewed the work that's been done so far, the answer to this question of have we got this covered, are we strong now, and do we have strategies for addressing research reproducibility? My answer really is that possibly we still have more work to do. And as we think about that work, I just want to call attention to the influence of our stakeholders, who I think have made a fairly robust and rigorous response to the concept and the weaknesses of perhaps our research progress. And so the stakeholders to our research data, our research inference, and our publications are, of course, the publishing agencies. They have a lot at stake for publishing the data that we generate and submit to them. The funding agencies, of course, who are funding our work, but other groups are also interested in research outcomes, and that would, of course, be the government agencies, professional organizations, private research organizations, and no doubt the public has a very strong stake based on public support of research and their high hopes for the research outcomes that we all seek to deliver. And as we think of those stakeholders, these data come from another paper from Friedman, who is an economist, and he looked at the cost of irreproducible research. So what he's conducting is research on research. How are we as researchers doing in our goal to provide solid biomedical research outcomes? And in this chart, he reports, based on his team's economic analysis, that half of the U.S. annual investment in research is deemed to be irreproducible based on their analysis. And that analysis was based on reviewing these pivotal research projects and trying to see if they could be reproduced based on what was provided in the manuscripts. And he provides in this table the categories that the problems were able to be generalized to. And so the categories of preclinical irreproducibility in this research were proposed to be problems with biological reagents and reference materials. So 36% of the research studies that they evaluated, they were not able to get enough information to be able to do another study and find and use those critical reagents. Study design was problematic in nearly a third of the projects. Data analysis and reporting, about an equal number, and then the absence of effective laboratory protocols to allow work to be reproduced. And when you look at those four categories, you will see that these data really guided the response of our stakeholders. And what you see on this slide is some indication of what the publishers and our National Institutes of Health as a funding agency, where their response to research irreproducibility has been focused. So in both of these, the publishers and the National Institute of Health have focused on what can we do now to help scientists improve those categories. So study design, methods, protocols. And when you look at the NIH response, which some of you may certainly be familiar with these already, this response has been in the last year, and they have requested that the application instructions from scientists for public funding need to be improved and focus directly on enhancing rigor and reproducibility. And when you look at their four strategies, that they're asking for additional details into the scientific premise, the hypothesis, the soundness of the approach. They're looking for more details and a expanded description of that experimental design. They'd like more details about relevant biological variables, again, the soundness of the science, and then the authentication of key biological and chemical reagents. And I think you can see that these topics work to address some of the issues that you saw in Friedman's analysis of contributors to research reproducibility. Now, on the publisher side, folks like Nature and Science, and they brought together other publishers, and they have also put interventions in place, asking scientists to deliver more in terms of materials and methods. They're incorporating the review of additional scientific statistical experts. So they're also looking at this soundness of the science and looking to improve. Now, this quote is from the NIH that indicating that, of course, these efforts will not be sufficient to affect real change in our climate that has demonstrated a problem with research reproducibility. And so I think that, again, the response of the publishers and the funding agencies, government agencies, and professional organizations have also made suggestions for what we as scientists should be doing to improve our success rate with our research. But a question I often think about is, well, what about research institutions and the scientists who conduct the research? What about us? There's no one really who cares more about our data and knows more about or should be driving our response to how data and research should be conducted. So this question is being asked by others. This is a Nature commentary, which I particularly like this diagram. And in the article, they're asking this question: what should institutions be doing to support their scientists and to support research reproducibility? And as you see on this diagram, we have on the far left the funders being held together with Band-Aids, and the publishers depicted by Nature there starting to crack the journals. And then what looks like a very beleaguered scientist who's just trying to figure out how can they do their work in a strong scientific culture environment while the robustness and rigor of science is kind of crumbling around their head. And then we see the institutions being depicted as being somewhat asleep in the wheel in trying to support the scientists in their effort to meet the needs of the public, the funders, and the journals, and of course meet their own needs for conducting strong research. And in this article, they call for what is depicted there as GIP, which are good institutional practices. So they make recommendations for what might be some institutional practices that could be incorporated as strategies for generating more robust research. So again, I'm going to take you back to this table and propose that when we think about sound scientific principles, these topics of study design, attention to bias, statistical subject matter expert expansion are being rigorously looked at by the scientific community and by our stakeholders. However, I would also propose that the second category of good quality practices that is so much needed is not being addressed or considered as strongly as it perhaps should be. And since both are critical for reproducible research, I think that it would be important for us to think about this. So if good quality practices do generate a critical gap in our work, what kind of practices am I talking about? So when I speak about good quality practices, I'm talking about at the point of data and metadata collection. Are we able to generate very reliable, transparent results throughout the life cycle of our data, from source data to archiving and sharing of data? Do we have records related to our equipment, the personnel who have supported the conduct of the science, our supplies, our methods, facility type records, or research records? What's the quality of these records? And I'm going to call your attention back to the Reproducibility 2020 paper, because the next slide refers to this area of that our data and metadata reconstruction are truly critical to research reproducibility. And what do we know about our ability to reconstruct our data and metadata as scientists? Now, this is the graph, and I'm hoping you can see this on your screen well, that comes from the 2020 paper. And what it shows is kind of the life cycle of our research. And as you look closely at it, it indicates quality checkpoints throughout that process. Some that in orange that are present, some that are rarely used, depicted in gray. And as you can see at the beginning, where we're coming up with a project idea, we have and then we go to funding, we certainly have a review process there because we're working hard on writing our grants. Grants are reviewed by subject matter experts. And when we go through to experimental design and protocols, we have some journal requirements, some institutional requirements that drive how we design our work. But when we actually move into the generation of the data where protocols and reagents and equipment is used, there's very little evidence of quality control points. And as you think about that, you realize that this is the area where the data and metadata are all collected. And it seems to me we have a true gap or a potential gap in quality control there because we don't currently have mechanisms beyond mentoring, which of course is a very strong proponent of research practice, but it varies from mentor to mentor, and we certainly won't. It doesn't generate the kind of records we need to reconstruct our data. So, what do we know? Remember, I said that I would like to tell you about what data do we have that illustrates how we're doing with our ability to reconstruct data and metadata. And these are the kinds of studies I'm going to share with you in the next 3 or 4 slides. This is a study of NIH-funded early career scientists in the United States who say they have engaged in the particular behaviors you see on your screen. It's a very robust response. Over 3,000 scientists answered this survey, and these were anonymously reported results. And remember that since these are NIH-funded scientists, they typically would be considered to have very strong mentors and strong programs. And as you see these behaviors, you'll see that 12 to 15% are concerned about their study design methodology, perhaps being influenced by pressure from a funding group. So this would be some of the issues associated with incentive groups and pressure groups. Inappropriate or inadequate design, 14%. Again, I hope being addressed by some of the approaches being implemented by our publishers and funding and training programs. The idea of dropping observations or data points based on a gut feeling that they're inaccurate. Again, this is an important practice, which many of us, I think, would recall reasons for doing this. And I think that the important thing here is documentation, again, about why we might want to drop data points. But it shows that scientists feel a little bit vulnerable about those procedures. But the one that I find really interesting is that about 30% of scientists mention that they feel that their record keeping is not what they'd like it to be, and that they recognize that inadequate record keeping may be a problem in their general research. That's a large number of scientists. Here's another study on the reproducibility of science, looking at the unique identification of research resources in the literature. So this group of scientists recognized that the inability to uniquely identify critical research resources is very important in terms of research reproducibility. And so they designed an experiment to look at the identifiability of research resources in the biomedical literature. So again, they went back to the published literature. They created a rubric for determining whether research resources could be identified so that another scientist could use them in their research and reproduce the work. Interestingly, 54% of those multiple studies across the biomedical platform of research, they looked at many different types of biomedical science, and they found that 54% of those critical resources were not uniquely identified in the publications. And this is, certainly influences the NIH's interest in having us do better at uniquely identifying our critical resources, such as antibody lines, cell lines, knockout mice reagents. Another study that you see on your screen, whose sample is it anyway? This looks at a widespread, what they call misannotation of samples in transcriptomics studies, where they looked at, based on the data in the papers, was there a match between the genomics data and the sex data that would be expected based on that data. And they identified apparently mislabeled samples based on the sex and the correlation that should be with the data in 46% of the data sets studied. Again, these are fairly surprising numbers because they have to do with record keeping. Another study looking at gene name errors being widespread in the literature. This is recognizing that there are problems with Excel and how Excel has default settings that may create problems in sorting data. And what they found were in the published literature, approximately 1/5 of papers that contain supplementary Excel gene lists contain erroneous gene name conversions. And I find these papers very interesting because these are fixable problems that may be addressed by improving practices. So basically, to me, this looks like some problematic record keeping. And I think that record keeping is a strategy that we could improve on with relatively low pain. And I want to make the point that even regulated research, so even research that is conducted under a quality standard, has problems with record keeping. The most common deficiencies that are reported in annual summaries of studies that have come before the FDA for audit, some of those most common deficiencies include inadequate record keeping, incomplete study records, inadequate or the lack of standard operating procedures. So this is a systematic problem perhaps in our science. And it appears to me that some of the long recommended research quality assurance best practices might help in this regard and could be a scientist-driven strategy for addressing research reproducibility. And I say that because quality assurance management systems are specifically designed to improve and maintain the precision and accuracy of a product and establish routine performance. In our world, the products that scientists produce are research data, the inference that we take from those data, and of course publications. And we can't forget that our other product is the next generation of scientists, the critical importance of our mentoring and the skills and the best practices we teach throughout our training programs. Now, in spite of the critical importance of data in our lives in the basic research world, whether that happens on an academic setting or whether that happens in a research organization, a private research organization, it's generally very true that quality assurance support and expertise is rarely found in the basic research settings. And I've found that to be true beyond academia. Where basic research is conducted, it's very rare that quality assurance best practices are there to support the scientific effort. Where you do find quality assurance is along the developmental pipeline, where we have basic research on the far left moving through the preclinical development, clinical trials, and the manufacturing environments. And where you see the quality systems come in is where research is subject to requirements by law. So under good laboratory practices, good clinical practices, good manufacturing practices, all of these are required by the Code of Federal Conduct for Research. So, and a big part of that is having a quality assurance unit there to look at and review and safeguard data. However, at basic research level, where we're looking at disease discovery and drug discovery and trying to come up with some good approaches to impact our health, there's no regulation in place. There's very inconsistent approaches to research, and again, all of them mentor-based. And this might be a place where we could voluntarily integrate some of the quality assurance best practices to support the hard work of those mentors. Again, we've been doing excellent research for many, many, many years, but we don't have the data reconstruction ability to prove that. So what I like about quality management systems, and why I think that they would be very supportive of basic research and the scientists that conduct it, is that quality management systems generate evidence and generate records that allow us to reconstruct the work, either in the future when we need to reproduce work, or throughout our ability to try to look at our data and respond to questions or respond to variability. So when you see this screen, you'll see that quality management system records, the typical types of records that are collected and generated, have to do with, are the equipment you're using working well? Is it well maintained? Has it been repaired? Do you know that it's measuring where you need it to measure? Has it been calibrated? Do we have records to show that our methods have been validated and that appropriate quality control is being used? And you go through this slide to see, I think you'll get the idea that when we look at these systems and collect good records, we really can stand up for the quality of our work. And it also allows us to do something we're not so used to in basic research, is the ability to continuously improve our work. So without a quality system, I'd ask the question of whether we fully support our scientists and the quality of their data if we do not have quality systems in place. And just have a look at these questions now as you think about the work you do. So do you feel like you would be able to feel strongly that the research data you are generating are original, accurate, contemporaneous, legible, secure, and unchanging, and can you reconstruct them? Is our documentation complete in our work? Do we ensure consistency in procedures and training through the use of routine standard operating procedures? And do we have training records that over time we'd be able to look back and say, yes, that person was trained to do that procedure, so if we have questions about the procedure, we don't think they have to do with the person that's conducting it. And then dare we ask about the maintenance of calibration of equipment, especially in our basic research settings. Have we given due thought and due investment to having strong equipment calibration support systems and maintenance data? And I believe that strategies really are needed to fill the gap here in our basic research settings. Now, I'd like to just say that research QA is not a new concept. It has been long recommended since the early 90s, and there are strong resources and effective resources available to scientists, and I just call your attention to a few of these. The World Health Organization document you see on the left in orange, Quality Practices in Basic Biomedical Research. The RQA, Research Quality Association in Great Britain, has a very nice, freely available quality systems workbook that is downloadable and can be used. And they also have a very nice little document called Quality in Research, which is available for about $10, which gives guidelines that can be incorporated. And then there's also a toolkit provided by the Michelson Prize and Grant Association for Research QA. And all of these promote the voluntary adoption of good research practices in basic research labs. And in addition to these are several publications. If you were to go and Google research quality assurance in basic research, you would find scientists calling for this as well since the 90s. And what's nice about quality assurance and about these resources is that truly these best practices can be adopted on an institution level, by small teams, or by individual researchers. I'll just give you a little bit more detail about the WHO handbook because it is such a nice reference or resource if you're looking for one. And you'll see here that I have some of their content about what they were trying to do. And as you see, they're basically trying to help non-regulatory scientists who are not regulated by law to incorporate some basic QA approaches and to help them produce credible research data and also promote the credibility and acceptability of their work through their strong record system, which we as scientists have identified, or research on research has identified, as a problem for us. And by doing so, we're now in a position to generate credible evidence which leads to trust in our data and confidence in our results. So how might this all come together? And basically, I think that we should be thinking about data and research quality at a more micro approach. What might we do to secure and safeguard the quality of our data? And we can do that by thinking about good research practices, the second tier of generating sound science. So what that requires is a real focus on data governance. By that I mean, what do we do to secure data from source data to when the data is generated from the equipment, when it's generated by a particular person, when what are the analytical tools we use to analyze these data. IT support in terms of data governance is a big issue. We need to think about our electronic data and how we're going to handle big data. We really need to support sound science and good quality practices. We need to focus on training and incentive system, and we need to incorporate some of these stakeholder strategies. So all of these issues really will contribute to data and research quality. So to sum up, this is a commentary that appeared in Nature in January 16, talking about that quality assurance, and what they mean by it may not be sexy, and I'm sure that this won't be a surprise to any of you viewers if you've experienced quality assurance. It does require attention to detail, additional records which need to be certainly collected by scientists and their staff, and an attention to the full data life cycle. So while this may not be sexy or may not be what we call rocket science, it definitely contributes to rocket science and supports rocket science. And so quality assurance really is becoming a crucial part of our lab life. And sadly, because as scientists, it's been long neglected in our training programs, we may not have the skills and the best practices as part of our research environments because they've not been part of them in the past. And so for that reason, I think it's important for us to think about integrating quality assurance best practices in our research culture in a way to promote best practices in sound science, to improve data reconstruction and research rigor, but also as a way to support our scientists. So from an institutional approach, I think that we need to be thinking about institutional centralized thinking with establishing and maintaining best quality practices to support the work we generate. And then it's very important to provide this critical training to address the gap in our training that we aren't incorporating QA in our training programs. And then lastly, because we've been able to generate evidence, we should be able to much more strongly demonstrate the quality of our work through our records and through our ability to continuously improve our work by paying attention to those records, and then readily be able to more quickly generate our publications because our records will be in stronger form, and and then also be able to communicate our quality systems when we're writing grants or submitting publications, to be able to say that we participate in a quality assurance system should make us stronger candidates for funding and publication. So with that, I'll stop my remarks and basically plant this seed that perhaps we could think more as scientists about how do we drive research best practices, what is our response to the research reproducibility issue, and how can we be more proactive to the concerns rather than reacting to the best practices that our publishing communities and funder stakeholders are laying out for us. What is our response and what can we do as scientists? Thank you so much.
Fabulous. We want to get right into the questions and input. So here's a reminder as to how audience members can communicate with us. Questions can be submitted via the Q&A button at the lower left. If we're unable to get to your questions due to time constraints, Dr. Davies will reach out to you via email. So let's take a look at our audience questions. Dr. Davies, our first audience member would like to know, Research QA is a new approach for basic research laboratories. What suggestions do you have for getting started with this in a research lab setting?
Is that truly you don't have to wait for an institutional approach. I would very much like to think that institutions will take this on board and will provide some support and will increase their training focus on research QA. But in the meantime, there are steps that you as an individual scientist could take. Let's say you're a mentor, you're a PI, let's say you're an individual scientist with you're a postdoc or you're a graduate student, and you would like to at least learn about what these best practices are or try to integrate some in your own work. All of this is possible by simply acquainting yourself with what these best practices are. And so my suggestion would be to go and download that WHO quality best practices in research and have a look at it. It gives you some real strategies. It gives you forms and templates that you could use, download, and use in your own work. So that would be my suggestion: is acquaint yourself with this body of knowledge, because if you're like me as a trained scientist in a PhD program, you may not have been introduced to some of these best practices under the umbrella of quality assurance. So the best place to start is acquaint yourself with the best practices and then see where you might be able to integrate some of them. And then secondly, to also talk with this among your peers within your lab groups, and in those conversations say, is there something we could do as our laboratory? Could we commit to some of these procedures and then get the benefit of them in ensuring the credibility of our work?
Thank you so much, Dr. Davies. It looks like we have one more great question from one of our audience members. If you were to select just a few of the research quality assurance best practices that are recommended, what would your choices be?
And this is a question that I've gotten before from groups that I've been speaking with about this topic. And basically I have two answers. One would be to look at the whole program and see what you're already doing and take advantage of the good practices you're already doing and just document them. The second thing is in order for this idea to work, it really needs to be science-centered and risk-based, right? So I'm not advocating that you put in a comprehensive program that is going to add too much work to your days. So what you need to do is make this risk-based approach. So if you ask yourself, what parts of my research cause me some concern or keep me awake at night? Am I worried? What am I worried about in terms of my data? What kind of data do I collect? For instance, if it's all electronic data, how do I know that it's not changing as it gets transferred into spreadsheets or stored places or generated within equipment? Or if you are someone who has multiple pieces to a research project, multiple people at different sites collecting data, you might be more concerned about controlling how that data is collecting. Are people doing things consistently? How do you know that people at site A or site B are doing things the way you are expecting it done? In that place, an easy place to start and an important place to start might be to say to yourself and your project, we're going to have a consistent approach to some of the routine practices. So this is what standard operating procedures are made for. SOPs are perfect for routine processes that you want to capture and make sure are consistently conducted. So sample collection, perhaps, if it's being done at several places, you would want to have an SOP for a standard operating procedure for how those samples are collected. And you would want to share that with everybody who's doing it and get training records associated with that SOP. So nailing down your routine procedures. One of the best, most important places to start would also be having a consensus-based approach to how you manage your data. All right, so where are you storing your data? What approaches do you have to Excel spreadsheets? Do you nail down your formula? Are data stored in a secure network? Can people edit or change your data unknowingly? Do PIs have access to seeing data in real time? So for me, I guess the most important first step would be sit down as an individual or a group and determine what your approach to data management is and make a standing operating procedure for that. But then, of course, I'm a great believer in all the other aspects. Look at your equipment, capture equipment data. Make sure you've thought about calibration and maintenance of equipment. If you're generating data from equipment and you can't show records of maintenance and calibration, the truth is your data can be darn near worthless because you can't attest to the credibility. And quality control, of course, is another big piece. Do you have quality control in your methods, within your equipment, throughout the research process? So that's probably too long of an answer, but there are risk-based places to start. Thank you for the question.
Dr. Davies, thank you, and that was a fabulous answer. And did you want to add to that with any final comments in your presentation for the audience before we close today?
This is new to the basic research field. I can tell you that it is being incorporated in several laboratories. We're working with laboratories at the University of Minnesota to help them. There are international approaches to helping scientists with this. There are societies, the Society for Quality Assurance, which has university specialty committees and people who are interested in how to integrate quality assurance into basic research programs. So I really encourage you, if I was interested in giving this talk as a way to plant this seed, and I encourage you to think about it and to not consider it as bureaucratic overreach by a regulatory sense, but think about this as a scientist-driven approach to standing up for the quality of that very important and precious data that you generate and the research that you're conducting for the greater good. Thank you.
Thank you so much for this informative presentation, and honestly, thank you for focusing on best practices of research. That is such an important topic in every field. I'd also like to thank LabRoots today for making today's educational webcast possible. Today's webcast will be available for on-demand until August 2017. Keep an eye out for an email from LabRoots alerting you when the webcast will be available for replay. We encourage you to forward this announcement to your colleagues who may have missed today's live event. Thank you for joining us, and we hope you come back next time. Goodbye.